welcome to this lecture. So, now I am going to summarize whatever so far we discussed. Okay, let us see, just to recapitulate what exactly happening, just to link all of them together. So, what happened? I mean, what happened in immune system? I mean, why immune system is there? To protect us from infection, or mostly from infection, right. So, what is happening? There is a first line of defense. Okay, so, there is a first line of defense, and in this defense, what we have? In this defense, we have many things. We have our skin, we have mucus, immediately they are taking care. Okay. So, if there is any pathogen, it is not so easy for the pathogen to enter into our system, right. So, what is happening? If any pathogen, if there is a cut, that I am just going to repeat exactly what is happening. If there is a cut, and in that cut, so there is a, suppose there are infections, suppose there are bacteria. What is going the first reaction happen? The first reaction is going to happen that there will be a inflammation, and that inflammation will, that inflammation will make a lot of difference in the immune system. That will actually cause or to inform the immune system exactly that what is going to happen. So, what are the cells there? What are the immune cells there? There are neutrophils, there are monocytes, right. There are other cells like eosinophil, basophil, they are mast cell. So many things are there, and from there, from the site of infection or tissue, I mean infection site, tissue or skin, whatever is there, from there this antigen or the pathogen will bring to the nearest lymph node. In lymph node, what is there? In lymph node, there are T cells, there are B cells. Okay. So, when this thing is going to come, if our innate immune system, which is supposed to take care at the very beginning, are failed. Okay. If the primary line of defense just breached, and then this adaptive immune response will come. So, this antigen is going to be presented by presented to T cells by different APC, antigen presenting cells, which are macrophage. We also write macrophage like M5. We have dendritic cells, we have B cells here also as antigen presenting cells. So, these and macrophage or dendritic cells, what they are going to do? Suppose I am talking about the macrophage. So, in macrophage, what is going to happen? What I just discussed, right, in the last lecture, is that the antigen will come and it going to be internalized, and suppose if I take this color as red, it will be internalized, and this internalized vesicle will be in the vesicle, that will be chopped into pieces, different pieces, which will go with MSC 2. So, ultimately it will be presented by MHC2. This is one thing happening, and during this inflammation, during this fact that inflammation, what is going to happen? Lot of cytokines are going to happen, cytokines, chemokines, which will bring all this thing, I mean all the different cells into the site. And what is this symptoms of inflammation? Just to recapitulate again, that is heat, pain, swelling and redness, right. These will tell you that okay, I mean if all four or any at least first three like heat, pain and swelling is there, you will see that inflammation is happening. Inflammation means there are infection is there. So, this antigen processed in case of external pathogen, and in case of internal or cytosolic pathogen, then suppose the dendritic cells are going to take care, and that antigen or the viral antigen are going to be presented by MHC 1. Okay. Both these dendritic cells and macrophage are going to lymph node. What they are going to do? They are going to release lot of cytokines. They are going to release lot of cytokines. What these cytokines are going to do? These cytokines are doing two things. One, they will control the macrophage and dendritic cells called autocrine regulation. That autocrine regulation will activate the macrophage, lot of molecule will come. Will come again when we will see the T cell activation, what are those molecules, co-stimulatory molecules will come. So, these molecules are newly expressed, which was not there in macrophage. So, cytokines will produce and they will activate itself. Okay. So, the cytokines both in case of dendritic cells and macrophage, they will activate itself. At the same time, the cytokines also activate B cell and T cell. So, first this macrophage is going to, it is better to this T cell part first. This both the dendritic cell as well as macrophage produce cytokines that is going to activate T cells depending on their activity. Like dendritic cell, the MHC1 will activate cytotoxic T cell, macrophage with MHC2 will activate T helper cells. So, the T helper cells, this macrophage, the T helper cells are of two type. One is going to make the Th1 response. That Th1 response again activate macrophage to kill the internal pathogen or intravesicular pathogen inside it, and there are one T follicular T helper follicular cells, which is you are going to learn in much more detail that this T helper cell is going to help the B cell. There is one very important thing is there in immune system, because there is always a possibility that either T cell or B cell can do the mistake, okay, because if there is no control. So, that is why both the activation of T cell and B cell, it is important that both the B cell and T cell should get two different signal. Okay. What is this two different signal? What is this two different signal? Two different signal means suppose this is the T cell, okay, this is the T cell. So, what is happening? This T cell is, this receptor is there. Okay. If any antigen binds here with presented with the MHC, this is the MHC molecule, this is macrophage. So, this interaction, this is TCR, this is TCR, this TCR and MHC, this interaction will give one signal. Okay. Suppose this is signal 1. This interaction is going to give a signal to the T cell that this interaction is happening. If this thing happen only, then T cell will not be activated, because there may by mistake it can happen, because so many because in lymph node what is happening, there are so many T cells, this is packed, and there are so many lymph nodes are there, this is packed. So, when one antigen presenting cell is going, it is interacting with many T cells. So, some weak interaction can happen any time. So, this is not going to activate the T cell. So, this interaction should stable for some time, and this interaction, this activity, I mean how this antigen is presented, let me make the different color. So, how this antigen is presented, how this antigen become here in on top of MHC molecule. This antigen is processed because it is already taken up, the whole bacteria was taken up into vesicles, and this vesicle is make this antigen into small pieces. This is presented by MHC2. So, during this process macrophage get activated. Activated macrophage is what they are going to do. Activated macrophage are going to release some small molecules. Okay. Release some small molecules. They are mostly cytokines and chemokines. In this case, it is cytokines. So, these also will activate. Okay. This will also activate. So, both this, if this is signal 1, suppose this is signal 2. Both signal is very, very important. Either signal 1 or signal 2 is not going to do any change in the T cell, because you imagine one macrophage is there and lymph node is packed. So, there are many T cells. Okay. There are many T cells surrounded by this macrophage, all are T cells. All are T cells. So, this cytokine is a molecule. So, if this cytokine released by the macrophage, it is not only specifically there, I mean there is no person is there that go and fit there. So, these molecules can go either this side or can this side. So, cytokines can go and bind to this T cell, but if this, that means if this is the signal 2, it is not going to do anything to this T cell, because this interaction signal 1 is not there. So, even it is going bind and giving the signal 2, T cell will not be activated. So, this is very, very important. So, both the signal simultaneously is required to activate the T cell, whether it is cytotoxic or it is T helper cell. Okay. Same way, if we see B cell, okay, if B cell is there, so let me change the color. If B cell is there, okay, so this B cell, this is B cell, all B cell has their receptor, looks like antibody, because they are basically the antibody molecule. So, this antibody molecule can bind the antigen freely, that antibody molecule can bind the antigen freely. So, they are attached to it. So, antibody interaction with the antigen. So, this red is antigen, this antibody interaction with the antigen also gives some signal, and this all the receptor, when the antibody is receptor, they have some cytoplasmic tail. Okay. So, as soon as this thing bind to antigen binds to the antibody or the B cell receptor rather. So, this is B cell receptor, it will also give a signal. Okay, it will also give a signal. So, this is again signal 1. So, this signal 1 can happen any time, because antibody B cell receptor can bind to many things. which is not that strong binding, or some self antigen is slightly similar and to the B cell receptor, may interact for a while for very short period of time, less efficient or less affinity, but this signal even if it is there, B cell will not be activated. Okay, even this is a very one more thing we will discuss, it is if this signal 1 or the interaction with B cell receptor and antigen happen only. So, if there is no other signal, then that B cell will be anergic. Okay. So, only signal 1 if happen, if this signal 1 is very strong, that means this interaction is very strong. If this thing happen, this B cell will be anergic. Anergic means they will not work any further throughout their life. Okay. They will be totally silent or irresponsive to immune system. So, they are basically out of the immune system. So, that non-specific interaction somehow it happened, that B cell will be gone. Okay. So, this thing is only this thing is not possible. So, what is going to happen? This thing then deactivated T cell. So, same antigen are presented by the macrophage, and if this is MHC2, then this T cell will be activated. These T cell activated T cell, I mean they are in lymph node, they are very close. Okay, it is not that much space like the way I am drawing. These T cell activated T cell will also release some cytokines, and these small molecule, okay, these cytokines will also act on this B cell. Okay. So, this will give a, because this is also there is a receptor where it will bind. So, this will give a signal 2. So, one signal will get from the antigen BCR interaction, another signal will come from this cytokine which will give the signal 2. So, in B cell activation both signal 1 and signal 2 is very essential. If this thing does not happen, then we will not see any B cell activation. B cell activation means what? B cell activation means this particular B cell will be converted to plasma cells, and in plasma cell what will happen? As soon as it become plasma cell, this B cell receptor BCR will be spliced separately, that we have seen in during the antibody diversity and antibody processing time. So, it was like initially it was receptor and the same antibody molecule will be produced which does not have any secretory domain by alternate splicing, that will release the antibody in the blood. Again this what kind of antibody, whether it is Ig, first antibody will be IgM, and I already told how it is switched and what IgM, IgD, the alternate splicing. Okay. So, this whole process is going to take time, and that is why the first thing. So, in this case when it is, when we are saying it is very simple and straightforward, it is not, because all this, so this antigen processing part we have discussed. Okay. So, when at the very beginning at the introductory class we have discussed all these things. So, the antigen processing, both the in case of viral antigen or tumor antigen or extracellular antigen or the pathogen, the processing. So, this one line thing antigen processing means, you know, there are almost 1 hour lecture what thing is happening. This T cell activation, like what is happening, we will discuss in next to next class, how exactly T cell happening. This B cell activation also will be discussed. So, this is the summary like what we have discussed so far. So, what we know? We know how this BCR generated, why we need so many antibody or BCR or so many antibody or TCR, because there are so many different pathogens. So, each one is responsible for a specific peptide. Okay. So, that peptide is generated from where? That peptide is generated from antigen. Okay. So, this what is this antigen actually now? What is this antigen? If we see, so suppose this is a protein. Okay. So, this is a protein, and this protein, this part is one epitope. What is epitope? We know that which part of the amino acid or protein is recognized by the antibody. Then it may be a different epitope. Clear? Again, this may be an epitope. What is the difference between this red, green and black? What is these three epitopes? Suppose this antigen, suppose this is one antigen, antigen has epitope 1, epitope 2, epitope 3. What is the difference? Difference is, I mean, if you see carefully, this is the 3-dimensional conformation. If you see this, okay. Now, if you make a straight line, suppose this is the primary sequence of the peptide. Now, what will happen? You will see red is here, okay. Then we will see green is here, but where is the black? Black you may find some are here, some here. Why? Because you see this part actually contributed by fold, okay, because two parts of the protein come together and make a structure which is recognized by the B cell or B cell actually, right. In T cell it is not going to happen. T cell is always one type receptor because it is chopped, but B cell receptor can recognize the whole protein. So, if this is the case, so then in case of 1 or epitope 1 and epitope 2, you see it is a continuous sequence. This case it is called linear epitope. But in case of epitope 3, it is generated because of a particular fold, particular conformation. If you denature it, it will not be there because that will be spread like this. This is called conformational epitope. Okay. Conformational epitope, because this is generated by 3-dimensional structure of the protein. Clear? So, now once this is a protein, it is not that one B cell will produce. So, once one T cell is activated, here, that T cell present, and in lymph node there is not only one B cell, there are so many B cells. Okay. These will continuously try which can fit into that, because in this case there is no question of here the B cell receptor can recognize the whole antigen, you do not need to chop it. Okay. So, what is going to happen? So, it is possible, I am taking the B cell again. So, it is possible that one B cell, this is B, say B1, can recognize this, another B cell, that is B2, can recognize this, another B cell, suppose B3, can recognize this. So, what is happening? One antigen entering into our system, brought to lymph node, presented to T cell, and B cell also can see. So, what is going to happen? One antigen can activate many B cells, each one will produce, I mean each B cell will, what will happen? They will activate, they will multiply again, many in number, all are identical, right. Here it will all be 1, there it will multiply again, there it will multiply 2. So, each one will, some of them will be converted to plasma cell, and each group of each cluster, basically each type of B cell will produce antibody. They are different, but can target the same antigen. One particular activated T cell or one class of T cell, what will happen after activation? After activation, what will happen? They will also multiply, right. If you see, if you remember that on side of dendritic cells they are multiplying. So, each activated T cells against that particular antigen, can come and activate each one, B1, B2, B3, because this is common. This particular set is common for particular antigen. So, this T cell can interact with different B1, B2, B3 cluster, can activate, and because that is why we need more T cells. So, each activated T cell will go and look which B cell is attached with the antigen. Okay. So, what will happen initially, it may the same, that is why it takes few days to process initial part, that is 7 days. One, it is activated, immediately what is going to happen? Once it is activated, it is activate many B cells. That many B cells may be of different type, that many B cells may be of different type, maybe B1, B2, B3, B4, because some antigen has only one epitope, some antigen may have multiple epitopes. So, more epitopes better for immune system, bad for pathogens, but what we will see, multiple type of B cell is are activated, they are produce variety of antibody, some are very good, some are less, but ultimately whole antigen will be taken care. Okay. And this is how the whole immune system works, and there are many other things, and then this B cell will that antibody will do the effector function, then complement activation, opsonization, neutralization, many other things, and then we will see the effect. Okay. So, so far whatever we have discussed how this diversity of this receptor of B cell generate, how antigen processed, and initially that you already know the innate immunity part, how this thing happen, and you know the complement system by now, and then how this thing generate B cell activation also you know. So, gradually, or if you do not know, you will know very soon, and ultimately the whole system attack. So, innate immune system will taken care, if it fails then adaptive will come, but once adaptive is there it will take care most of the infection, and that is why we do not see ourselves sick all the time. Even we are living in an environment, everywhere microbes are there, our water, our air, whatever we are touching, everything full of bacteria, even our skin, so many bad bacteria are there, but we are still safe because our immune system is working.